And we can tell that it's a muscle fiber because we can see the organelles inside of it. And if we look over here to the side, we know already that that muscle fiber is made up of myofibrils. So each one of these circles would be one myofibril. Here's a second, here's a third. Notice the little dots. Remember that myofibrils are made up of myofilaments. So each of these dots are those thick and thin myofilaments. We can see those myofilaments a little bit better if we come over here on the side. So can you see them right here from this, the thick and thin? Remember in our lecture we learned about the light, dark bands? So that's what we see right here. Can you see the dark band and then the light, the dark and then the light? Recall that in the middle of the light band is the Z disc. So if we look at this light band in the middle, here's a Z disc. This must be a Z disc right here. So from here to here must be one what? Sarcomere. From here, the middle of this light band to this middle of this light band must be one sarcomere. So if on the practical exam I put a little piece of tape like that and say identify this entire structure from here to here, it'd be a sarcomere. Also from this side view, we can see some of the organelles of the muscle fiber. This right here is a nucleus. Here you can just barely see these mitochondria right here. And then over here on this side, you can see these blue and beige structures. The blue structures represent what?
T-tubules.
T-tubules, very good. And then on either side, can you see this network right here? That's the sarcoplasmic reticulum. Recall that sarcoplasmic reticulum is just the fancy name for the smooth endoplasmic reticulum, where the calcium ions are stored. Specifically, the calcium ions are stored in this swollen region of the sarcoplasmic reticulum. That's called the cisternae, or terminal cisternae. Remember the triad in lecture 1, 2, 3? The two cisternae and then the T-tubule. So that's what is represented right here. Notice how it spreads all around and throughout our myofibrils. Because in a little bit, next time in lecture, we're going to talk about how the calcium ions are released and they're going to move those sarcomeres right here that we're looking at. Then if we come over here to this side, we can see a little bit more detail. So between these myofibrils is this beige area, and that would be the sarcoplasm, the cytoplasm of this skeletal muscle fiber. Those are mitochondria. And then in beige right here, going all the way around, that would just be our phospholipid bilayer, which is called the sarcolemma, the fancy name for the cell membrane, the plasma membrane of this muscle fiber. Notice right here that that plasma membrane becomes folded. Do you guys remember the name of the folded sarcolemma? What was that called? Motor what? Motor end plate. That was motor end plate right there. And in this region, that's where we're going to have a whole bunch of receptors for acetylcholine, right? This is where we're going to be able to get what we're going to call our local potential is in this region right here. And then this gray color that we see over here, that's outside of the plasma membrane, that, if you look at it from the back, that is abstract art, areolar connective tissue. That's our endomysium. Remember how you have connective tissue that surrounds each muscle fiber. And then what you see over here on also on the back, in this region, they took off they have right here this kind of splotches. And those splotches represent the synaptic knobs of the neuron that's sending the command to the skeletal muscle fiber to contract. So we know that it's that because if we look over here on this side, let me turn this just a little bit right here, and we move it this way. Can everyone see this circle right here on this? That circle right here is, and this right here, this is an axon from a neuron. And this is what we're going to call the myelin sheath that surrounds the axon of the neuron. And so it comes and then it spreads out and it forms that synaptic knob. So for some of you that might be thinking like, what is that? What are you talking about? Let me show you. In this model, they had to fit it, keep on that region. In this model, they had to fit it so that it could sit on the shelf. So what they did was they took a neuron. Does everybody recognize this as a neuron? Does everybody recognize that as a neuron? Can you see the end of the neuron right there with the axon surrounded by myelin sheath? Okay, so imagine if I could take this neuron right here and hook it up right there, I'm going to be able to send a message, right, to that muscle fiber. But they couldn't do this because it wouldn't fit on the shelves of most schools. So they just chopped it off right there. And that's why we only just see the very end of that axon, and then it's spreading out into a neuron. And then one more time, just to reiterate what that looks like on here is, or actually let me point out one thing and then I'll do a drawing. So if we look a little bit closer here to this right here, this in blue is the end of a synaptic knob. These little circles that we see are synaptic vesicles filled with acetylcholine. And then this tiny space right where I have my probe, all of this in between the sarcolemma and the end of the synaptic knob, that's called the synaptic cleft. So on the test, if I put it on the beige, it would be sarcolemma. If I put it in that space, it would be the synaptic cleft or synaptic gap, we can also call it. If I put it on there, that would be, you know, from here all the way out, that's the synaptic knob. Those are mitochondria, but those little circles are synaptic vesicles filled with acetylcholine, as we'll soon learn about in a little bit. And then just to reiterate on there, I'm going to turn the lights on now, we're good with that, this is what I'm trying to show you on that fiber, is I'm trying to show you this. Hold on for a second. And that is, imagine, here's my neuron right here, right, with its cell body. And it goes all the way down. Let's turn that off in just a second. And it goes all the way down like this. Here's the nucleus. These are the dendrites, cell body. This is the axon. The axon at the end always branches. And it branches into these structures that we're soon going to call synaptic knobs. They look like that, correct? And these synaptic knobs then innervate, they touch our skeletal muscle fiber. So right here, see the end of the knob and that tiny little space? That tiny little space is the synaptic cleft, and then this would be a, you know, motor end plate right there. And that's what they're trying to show you here on this model, is that little folded, you know, motor end plate, and then the synaptic knob, you know, innervating right here. This is just a little blow up of this little piece, you know, of this muscle fiber and this synaptic knob. Okay, and they show you the little axon. You know, that's just the end of it right there. Okay, so any questions on that one? You guys good on that? Okay, do you guys want me to walk through a few of the muscles? I think that was the hardest. I wanted to get the hardest